DNA replication. All right, we're going to start off just really hammering home exactly what 5' and 3' mean and a couple different things they can mean. We're going to go through the entire mechanism, and we're briefly going to talk about some, drugs. All right, so here's carbon number 1, carbon number 2, carbon number 3, carbon number 4, and carbon number 5. Carbon number 5 can be attached to a phosphate, and carbon number 3 can be attached to a phosphate. If we, look at this nucleotide A, B, C, and D, right? This is a tetranucleotide structure. We see that for D, 3' is not really bonded to anything, not to any other nucleotides. And here we see that for 5, or nucleotide A, it's the 5' that's not really connected to anything. That is why this is the 5' side, and this is the 3' side. Again, we have a 5' end and a 3' end, and now they run anti-parallel. So the thing that they're attached to, the 5' end will be on the bottom, and the 3' end will be at the top. To really super duper hammer it in, think of like Georgia and Florida, right? So here we have Florida, here we have Georgia, and here we have New York City. So Georgia is north of Florida, and yet it is south compared to New York, right? So something can be north and south at the same time. Similarly, if we look at a 5' and a 3', and again, all this the absolute North Pole, what this 5' means is just that somewhere here we have a 5' carbon on a ribose that's not attached to another nucleotide, and here we have a 3' the 3 carbon on a ribose that's not attached to a nucleotide. That's where we get the absolute north and south pole. But again, things can be relative. If you pick this little position here with respect to this end, we can call this the 3', and with respect to this end, we can call this the 5'. So it's very important, I think, to understand that relative 5', 3' directionality. So whole mechanism. Helicase shows up, starts unwinding. Boom. Look at that. We're going to watch that again. That is some cool ass graphics. All right. So now we have these single-stranded binding proteins. So DNA is negative, so you can assume that these are super positive. And why do they bind here? It's because if they don't bind, everything, these things are going to try to start, becoming a helix again, right? They want to reanneal, and we don't want that to happen. So the SSBs prevent reannealing. Primase is going to go ahead. Now, anytime, there's only one thing I can think of that goes 3' to 5'. And so pretty much when in doubt, things are always laying down always laying down RNA or DNA in the 5' to 3' direction. So notice if this is 5', then I would say that this is 3', right? 3' and 5'. And so what the primase is going to be laying down is 5' to what we would call the 3' direction. And that's exactly what we see. It's going to lay down in the 5' to 3' direction. All right. I always thought of DNA polymerase as being kind of a diva. It's like it needs a red carpet to start off, right? I won't even start doing my job unless I can walk off of a red carpet. And RNA is often depicted as red. So I'm seeing the DNA polymerase only starting at the end of the red carpet, the red RNA primer. This is going to be really important later for things like telomere. So DNA polymerase is going to extend 5' to 3' in that manner. As helicases, we can see moving to the right. So it's DNA polymerase 3 in bacteria. It's epsilon in eukaryotes, and it's so we have a leading strand and a lagging strand, which we'll get to. I always thought of Delta Airlines. You might have a jet lag, or that's how I remembered it. So the helicase is moving to the right, and the strand that's being the new strand that's being synthesized is moving in the 5' to 3' direction. So in other words, it's moving down here. It's sort of chasing the helicase. It's moving in the smart direction, right? That means it can just continuously run chasing the helicase. We're going to have the lagging strand, which is going this 5' to 3' direction. It's going to be moving in the opposite direction. So it's kind of stupid. It has to keep stopping and starting. So let's see exactly what I mean. Smart leading, boom. Look what the stupid. The stupid has to get up, run all the way around, get another primase, lay down, and do that again. It constantly has to keep doing that. It's just a little bit silly. So this is just a much better picture of the leading and lagging strand. The DNA is unzipping because of the helicase in this direction. So this can just follow it forever and ever and ever without having to pick up. But this guy constantly has got to go, oh, crap, oh, crap, oh, crap, oh, and it's just like a real hassle. Notice as well that every single one of these replication events is starting off of a little red RNA primer right there, etc. This is a bubble. This is going in both directions. We've only been depicting one side of this, where we have a leading strand here and then a lagging strand on the other side. But one man's lagging is another man's leading, right? So on this side, we have another helicase going this way, and a helicase going this way. And so now this is actually being the smart one on this side, chasing the helicase. So it depends. On one side, you're the leading, one side, you're the lagging. Okay, so here we have Okazaki fragments. So we had an RNA primer. So the DNA polymerase, which made this right here, it can't go, it's got to stop when it gets to the primer. So this is a little chunk, and this is a little chunk. And those are our Okazaki fragments. Now RNAs, so Okazaki fragments, RNAs can now cleave the RNA, kaboom, get out of there. And now DNA polymerase 1, so it's important, DNA polymerase 1 is replacing the Okazaki fragments. Polymerase 3 is laying the main track. This can just go ahead and moves 5' to 3' like everything, and it can fill in most everything. But notice we still have these little gaps, right? So DNA ligase is going to come in here and close those gaps. SSBs were here the whole time. It just was too lazy to draw everything. So they're just always there trying to prevent reannealing. Ahead of the helicase, we have topoisomerase, or called gyrase in bacteria. It relieves tension. What tension? Well, if you have a lanyard, or you have like a bit of string that you can twist up into a double helix, twist it up into a double helix like a smia, and then just start to like unpeel it like this. And what you're going to see is you're going to induce all of these tensions and coils. And if you do it too much and it's fragile, it might break. So the idea is that if you had topoisomerase, you would keep it nice and steady and prevent all of this. So there's two types of topoisomerase 1, topoisomerase 2. Number 1 cuts, you can see this little itty baby 1 cut right there. And here I can see that I've cut once and I've cut twice on both sides. So that makes sense. The 2 cuts the 2 strands. And names you should recognize are toposide, teniposide. So I always thought of like there's 2 sides to every story. And then Tekken, I always thought of Tekken as like the number 1 video game of my childhood. So Tekken, the stupid fighting game from the 90s, is topo 1, silly. Because they're inhibiting topoisomerase, specifically in eukaryotes, these can be used as chemotherapies. So cancer cells dividing too much, let's fuck up its ability to relieve supercoils and tensions when it's dividing, which it does a lot of. You also have fluoroquinolones, though, like ciprofloxacin, and these can inhibit bacterial gyrase. So these are going to be antibiotics. This is just a nice summary. So we're going to lay down a 5' to 3' primer. Boom, we have it right there. We're going to extend with DNA poly 3, which is going to chase the helicase in this direction. Here we have topoisomerase. This is the leading strand. It's chasing the helicase. But then we also have primer here, and then we have DNA poly 3 and another one, DNA poly 3, and that's why we have these 3 Okazaki fragments. But eventually we don't want the RNA, so then we have we're gonna get rid of it with RNA. So we're gonna try to fill in the things with DNA poly 1, and then we're going to ligate those Okazaki fragments together with DNA ligase. And this is all happening on one side. The leading and lagging will flip on the other side of the replication bubble, as we see there.